Parkinson's is caused by missense mutation, which means that a single base is changed in a single base pair. Parkinson's mutation occurs in the GIGYF2 gene, where a single C is changed to a G. This mutation is located on the second chromosome on the Q arm at the 37.1 location. It causes Parkinson's, a human disease that causes imbalance, joint stiffness, slow movement, and tremors. The current treatment is a drug called levodopa. First, we have to study the mutated sequence by PCR. We can get the original human DNA sequence using the AVA2 restriction enzyme, which can cut at two places, shown here. First, the DNA primers line up against the matching sequences on the denatured DNA strands. Then, a Taq polymerase will make the sequence of about 70 base pairs and amplify the DNA. This will continue for many cycles, until there are millions of copies in just a few hours. Now, once we have studied the mutated sequence, we can correct it in two ways: CRISPR and gene therapy. CRISPR will first have a target DNA and a second RNA component bonded to itself. The target RNA will look through the DNA for a matching sequence based on the PAM sequence AAC. This is called targeting. Then the sequence is found and the RNA primer bonds with the RNA. This is binding. After this, the DNA is cut 3 base pairs upstream of the PAM sequence. This is cleaving. Finally, the fixed gene is fixed through the HDR process and it is rebound to the DNA sequence. This is DNA repair.
We can also use gene therapy through a plasmid called PUC18. We can take out the correct part of the DNA after amplifying it through PCR using the AVA2 restriction enzyme, and cut the PUC18 plasmid using the same enzyme and insert the correct gene into the plasmid. The DNA and plasmid restriction sites are shown here. The PUC18 plasmid is cut, and then the human DNA is inserted with the correct sequence. This plasmid can be put into the E. coli cell, which delivers it to human cells that replicate the correct gene. These properties can be used to see if the DNA sequence is correct based on electrophoresis. A double digest with AVA2 and BSMAI can show the difference to us. All the restriction sites are shown here. And here's the final electrophoresis. Notice the difference between the 731 base pair strand and the mutated DNA cut with the AVA2 and the 704 base pair strand and the normal DNA.
Thanks for watching.